Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lipid chemistry lecture, I will be going over classification of fatty acids and any applied clinical aspects related with the classification of fatty acids and different fatty acids that I am going to talk about. Let's get into the details. Note that the lipids are heterogeneous molecules. These are the esters of alcohol along with the fatty acid. So along with the fatty acid, lipids will, they can be carbohydrate part, nitrogenous base can be attached. So different molecules can be found in lipid molecules. So some of the lipids of, for example here is they are fats, oils, waxes and any related compounds like cholesterol. So as per the Bloom's criteria, so lipids are insoluble in water and soluble in non-polar solvents. Because lipids are generally, they are non-polar in nature. That's why they are soluble in non-polar solvents but not soluble in water. So some of the lipids that I would like to talk here, especially which contains different types of fatty acids, is like triacylglycerol. You know, triacylglycerol, it contains glycerol which is attached with three fatty acids. These three fatty acids can be different type. It can be a long chain fatty acid, short or medium chain fatty acids. I'm going to talk about what exactly is a short chain fatty acid or a medium chain or a long chain fatty acid. So overall, triacylglycerol, it is a storage form of energy in our body and especially stored in the adipose tissue. And also note that triacylglycerol or the adipose tissue that stores triacylglycerol, it acts as a thermal insulator, thereby it will kind of maintain our body temperature. So waxes, they keep skin lubricant and waterproof because they will repel water. Phospholipids are the membrane components. These are the phospholipids contain phosphate and glycerol which is attached to the fatty acid. And they can act as a detergent, surfactants, and also they will provide second messengers. And the important phospholipid that will provide second messenger is phosphatidylinositol 4,5 bisphosphate. And then sphingolipids. Sphingolipids are also component of our cell membrane and especially the nervous tissue and myelin sheath. These are the type of sphingolipids it contains. And then sterols. Sterols are biologically useful compounds such as bile acids, steroid hormones, sex hormones, vitamin D, and also sterols can be part of our cell membrane. So like this, we have different lipids which contains different types of fatty acids and they have a variety of functions in our body. So this table is just not an exhaustive list of biological functions of lipid, but some of the important biological functions of some of the lipids in our body that I have enumerated here. Coming to fatty acids. Now the fatty acids are, these are carboxylic acids with aliphatic hydrocarbon chain, which the chain length can vary as it is shown here. So there is a carboxylate group here, carboxylic acid group, which is COO minus on this side. And then that will be number 1 carbon. That's how we number the carbons in a fatty acid. You always start from carboxyl carbon, that's number 1 carbon, which is a deprotonated here. COOH is deprotonated into COO minus. And that's because the pKa of this carboxylate group attached to the fatty acid is 4.85. Now the pK is 4.85 and pH is 7.4. That means we are dealing with pH more than pKa. That means there is a deprotonation of this carboxylic acid group into carboxylate. So that carboxylate group here is a number 1 carbon. That carbon is number 1 carbon. And then we'll start numbering going towards the methyl end. So this side is the CH3, all the way other side is the methyl end. So number 1, number 2, number 3, number 4, number 5, you just go on naming the carbons like that. So you always start from the carboxyl carbon as number 1 carbon. And then note that the carbon which is besides carboxyl carbon, that's number 2 carbon. Number 2 carbon, sometimes it is referred as alpha carbon and then number 3 as beta, number 4 as gamma carbon. So alpha, beta, gamma carbons you should be aware of. Second carbon is alpha carbon, third carbon is beta carbon, and the last carbon in a fatty acid, that is the methyl group CH3, very last carbon, it is a omega carbon. It is referred as omega carbon there. Okay, also sometimes referred as nth carbon. Okay, so that's how a fatty acid, every fatty acid, it has got a carboxyl end and also methyl end. So carboxyl end is COO minus and the methyl end is CH3. Now the polar carboxylate group and the non-polar hydrocarbon chain will make this fatty acid amphipathic, because the carboxylate group, which is on the right side here in this fatty acid, this is polar, this is the only polar part in a fatty acid, and rest all is a non-polar hydrocarbon chain. So depending on the length of this hydrocarbon chain, so it will determine whether the fatty acid is soluble in the blood or not. If the hydrocarbon chain is smaller, like if it is less than 12 carbons in length, so this carboxylate group like COO minus, it will keep the hydrocarbon chain relatively soluble in water. As the hydrocarbon chain length increases, like more than 12 carbon, if it increases, then this particular COO minus won't be able to keep the long hydrocarbon chain polar. So it is becoming non-polar at that time. So it becomes not soluble in water. Okay, so you got to remember that point. Now, based on the number of double bonds in a fatty acid, so we can classify fatty acids into saturated fatty acid if there is no double bonds in a fatty acid, or unsaturated fatty acid if there is one or more double bond in a fatty acid. Now the first number 1 fatty acid that is shown here, it is a palmitic acid which has got 16 carbon, and note that there is no double bond in this palmitic acid. That's a saturated fatty acid. Now look at number 2. Number 2 fatty acid that is shown here is oleic acid, and oleic acid has got 18 carbon, that is C18. C18 indicates it has 18 carbon, and then there is number 1 written here, colon and number 1. Number 1 indicates it has got single double bond. So that is referred as mono unsaturated fatty acid. One unsaturation is there in this fatty acid, mono unsaturation. And then look at number 3. Number 3, it's a linoleic acid. Linoleic acid has got 18 carbon, but you see 2 double bonds in this fatty acid. That's why it is written 2, number 2 here. C18 colon number 2. Okay, so that means it is a polyunsaturated fatty acid. More than one double bond, we simply call it as polyunsaturated. So that means based on the number of double bonds that are present in a fatty acid, if there is no double bond, you call it as a saturated fatty acid. If there is one double bond, you call it as a mono unsaturated fatty acid. If there is a more than one double bond, you call it as polyunsaturated fatty acid. Okay, now depending on the number of double bonds and also depending on the number of carbons present in a fatty acid, so it can this particular table is giving you different names, common name, systemic name, melting point, and also percentage of occurrence in the nature. Let's get into little more details on that. Okay, so first we are looking at saturated fatty acid which do not have any double bonds. So number 12, carbon number 12, C12. So the 12 carbon fatty acid, the systemic name is dodecanoic acid. Dodecanoic acid means deca means 10, do means 2, so that means 12 carbon. Usually it is called as lauric acid. Lauric acid, melting point is 44.2 degrees centigrade. Frequency of occurrence is less than 1%. Now like that, if you go on looking at it, then we have a 16 carbon fatty acid which is hexadecanoic acid, which is common name is palmitic acid. Usually we use common names most of the time. Generally we don't use systemic name, but you should have an idea. What is the meaning of hexadecanoic acid? Anoic acid, that means no double bond. Hexa means 6, deca means 10, so 16. 10 plus 6, 16. That's palmitic acid. The melting point is 63.1 and occurrence is 23%. That means frequency or the abundance of palmitic acid is more here. So, and then we have 18 carbon fatty acids, stearic acid, melting point 69.6, occurrence is 6%. And then arachidic acid melting point is 76.5. It has 20 carbons. So, point here is, as you observe, as the number of carbon length, the hydrocarbon length increases, going from 12 all the way to 20. Look at what happened to the melting point. Melting point is also increasing because 20 carbon has got a melting point of 76.5, whereas 12 carbon fatty acid has a melting point of 44.2. That means as the hydrocarbon length increases, it needs higher temperature to melt this particular fatty acid. So, increase in hydrocarbon length increases the melting point. Make sure you remember that point. Okay. And palmitic acid is the most abundant fatty acid that is occurring in the human body. Now, coming to unsaturated fatty acid, the nomenclature, systemic name, common name, melting point, and also frequency of occurrence in the body. Now, let's look at the way these unsaturated fatty acids are named. Let's start from the oleic acid. Oleic acid has 18 carbon. That's our very first number that you see is 18. That's the total number of carbon. And then the second number you see is the number of double bonds present in that fatty acid. So, 1 double bond. So, 18:1. And then we have delta 9. That means it is a position of double bond, 9th carbon atom. So, the double bond is between 9th and the 10th carbon atom. That's what is the position of double bond. So, what is the systemic name? It is octa-deca. Octa means 8, deca means 10. So, 8 plus 10 is 18. That's 18 carbon. And enoic acid, any fatty acid that has a double bond, we name it as enoic acid. No double bond means it is written as enoic acid. If there is a double bond, we write it as enoic acid. That's the difference there. Make sure you know that. So, common name is oleic acid. Melting point is 13.4 and occurrence is 50%. Octa-decaenoic acid, oleic acid, 50% of total occurrence. Now, look at 18:2 and delta 9:12. That means this particular fatty acid has total number of carbon 18. It has 2 double bond. And the double bond is between 9th and 10th carbon atom, 12th and 13th carbon atom, 9-12. That's what is the meaning of 9-12. So, linoleic acid is the name and melting point is -5. So, like that, if you go on, we have 24 here. Total number of carbon is 24 double bond and they are present at 5, 8, 11 and 14. And arachidonic acid is an example. -49.5 is the melting point. So, if you observe carefully, as the number of unsaturation increases, as the number of double bond increases, going from 18:1 to 24, you look at the melting point is decreasing. That means higher the unsaturation, decrease in the melting point. That means it needs less temperature to keep the fatty acid soluble or to be present in a liquid condition. So, higher the unsaturation, liquid is the fatty acid. Higher the saturation, fatty acid tend to be in a solid form. Higher the unsaturation, fatty acid tend to be in a liquid form. Remember that. Okay. So, that's about some of the examples for unsaturated fatty acids here. Coming to a nomenclature, which I already briefly explained you earlier. So, the carboxyl carbon, that carbon we put it as number 1 carbon there. And then we go all the way towards the methyl end. So, this 18 carbon, that's CH3 here, that's the methyl end carbon. So, the second carbon, which is besides the carboxyl carbon, is referred as alpha carbon. Third carbon is beta carbon and the fourth carbon is gamma carbon. Alpha, beta, gamma, delta, epsilon, you can go like that. And the last carbon, that's a methyl end here, that is referred as omega carbon. So, this is how we basically number carbons present in a fatty acid. So, always remember, we all start from the carboxyl carbon in a classic numbering and go all the way towards the methyl end. So, and then we have alpha, beta, gamma, delta, epsilon carbon, and the last carbon in a fatty acid is omega carbon. So, now the stearic acid is the most abundant fatty acid in nature, basically coming from plant source. Coming to some of the information on saturated fatty acid. Now, the fatty acids with no double bond between carbon atom is referred as a saturated fatty acid, as we have already seen. And the hydrocarbon chain length can vary from one fatty acid to another fatty acid. And the most simplest saturated fatty acid is acetic acid, which is simply it has just 2 carbon. Whereas the stearic acid is the most abundant fatty acid in nature. So, which has got 18 carbon and no double bond. Okay. Now, sources of saturated fatty acids like butter, cheese and animal fat. So, animal derived lipids or fats are basically sources of saturated fatty acid. Remember that. And also consumption of saturated fatty acids, they positively correlate with atherosclerosis and cardiovascular events, because higher the consumption of saturated fatty acids have been correlated with higher incidence of cardiovascular events because of atherosclerosis process that will be going on. Now, look at acetic acid. Acetic acid has 2 carbon here. So, it is a major end product of carbohydrate fermentation, especially in ruminant animals like cows. Then butyric acid has got 4 carbon. So, it in certain fats in small amounts, present in small amounts, especially in butter. It's an end product of again carbohydrate metabolism by ruminant organism. Valeric acid is having 5 carbon, caproic acid has 6 carbon. So, like this, smaller hydrocarbon length chain fatty acids these are some of the examples there. Now, look at lauric acid. Lauric acid has got 12 carbon, myristic acid has 14 carbon, palmitic acid 16 carbon and stearic acid 18 carbon. So, some of the sources or occurrence is given here. Lauric acid, you can see it is particularly rich in coconut oils, spermaceti, cinnamon, palm kernel, laurels, butter, coconut oil. So, it is all rich in lauric acid. Whereas myristic acid, 14 carbon fatty acid, present in nutmeg, palm kernel, coconut oil and butter. Palmitic acid is common in all animal and plant fats. So, also these are the fatty acids are formed in cecum of herbivores, and to lesser extent it can be formed in colon of humans. Coming to unsaturated fatty acids. Unsaturated fatty acids, generally they will have more than one double bond, and they can be further divided into, depending on the number of double bonds, unsaturated fatty acids can be divided into monounsaturated fatty acids and polyunsaturated fatty acids. Now, the monounsaturated fatty acids, they contain one double bond and generally they are viscous and liquid at room temperature. Examples like olive oil, then we have canola oil, peanut oil, safflower oil, sesame oil, or even avocados, peanut butter. So, nuts and seeds, generally they contain higher quantities of monounsaturated fatty acid. Coming to polyunsaturated fatty acids, sometimes referred as polyethanoid or polyenoic acids. So, they will contain more than one double bond, and because of the higher number of double bond, they are less viscous and generally they will be liquid at room temperature. So, their consumption, they are negatively correlated with atherosclerosis and cardiovascular events. It's kind of exactly opposite to what we have seen with saturated fatty acids. Example for sources of polyunsaturated fatty acids, we have soybean oil, corn oil, sunflower oil, and also some of the fatty fish like salmon, mackerel, herring, trout. All of them, they contain higher amounts of polyunsaturated fatty acids. Remember that point. Now, look at the figure here, which is showing you how a polyunsaturated fatty acid can exist. Predominantly, wherever there is a double bond in an unsaturated fatty acid, usually the double bond is in cis configuration. That means the fatty acid tail, after the double bond, it changes the direction of a fatty acid tail. Whereas some of the unsaturated fatty acids, they can have double bond in trans configuration. And that means the fatty acid tail do not change the direction after the double bond. It's like a straight chain, just like saturated fatty acid. So, the trans double bonds fatty acids, they look like a saturated fatty acid. That's why food that contains higher amounts of trans unsaturated fatty acids, they are equally like that of saturated fatty acids, and they correlate positively with atherosclerosis and cardiovascular events. That's why consumption of trans fatty acid is not good for health. Now, an unsaturated fatty acid, as I was saying, it can be cis or trans configuration. Trans configuration fatty acids can also be done by hydrogenation process or hardening of saturated fatty acids in the manufacturing of margarine. So, that means a polyunsaturated fatty acid coming from plant source or something, if it is hydrogenated, where a cis configuration fatty acid can be converted into trans configuration fatty acid, thereby a liquid fat oil can be hardened into margarine. Okay, that's how the trans fatty acid can be done. So, small amount of trans fats can come from ingestion of ruminant fat. So, increased consumption of trans fatty acids are associated with increased risk of cardiovascular diseases and diabetes mellitus. You got to be aware of that part. Okay, now let's classify fatty acids based on the number of carbon atoms. So, based on that, we have a short chain fatty acid, medium chain fatty acid, long chain fatty acids, and very long chain fatty acids. Now, the short chain fatty acids, they contain carbon less than 6. So, any fatty acid that contains less than 6 carbons are simply referred as a short chain fatty acid. Generally, short chain fatty acids are derived from microbial fermentation of carbohydrates in the intestine. And example for short chain fatty acids, we have acetate, which is a 2 carbon fatty acid, propionate is 3 carbon, butyrate is 4 carbon. So, 2 to 4 carbon here, acetate, propionate, butyrate. So, they are all short chain fatty acids, and these fatty acids are generally taken up by the colonic epithelium for its own energy needs, or they can go into metabolism, and in fact they will control our energy metabolism, metabolic pathways. Coming to medium chain fatty acids. Medium chain fatty acids, they contain 6 to 12 carbon atoms. Example, we have caproic acid, which is a 6 carbon fatty acid, and then caprylic acid has got 8 carbon. Then we have capric acid, 10 carbon, and lauric acid is a 12 carbon medium chain fatty acid. So, basically medium chain fatty acid, they contain carbons anywhere from 6 to 12 carbon. That is what is classified under medium chain fatty acid. So, coconut oil is rich in lauric acid, and note that both short and medium chain fatty acids, they are water soluble. So, that means they can be directly entering into the portal system after their absorption from the intestine and carried to the liver, and in the liver they can be oxidized for energy purpose. So, generally they don't contribute to storage form of fatty acid in the adipose tissue. Now, long chain fatty acids are usually containing carbon more than 12 and less than 22 carbon. That's what is a long chain fatty acid. Example, we have palmitic acid, stearic acid. Palmitic acid is 16 carbon, stearic acid is 18 carbon, and the most abundant fatty acid that is synthesized in our body is palmitic acid, while stearic acid is the most abundant fatty acid that is available in the nature. Coming to very long chain fatty acid. Very long chain fatty acids, usually they contain 22 or more than 22 carbons. Generally, very long chain fatty acids are found in neuronal tissue, and whenever there is a turnover of these fatty acids, so initially they are oxidized in the peroxisomes and later they will be oxidized in the mitochondrial matrix. So, this is about a little bit about classification of fatty acids based on the number of carbons as short chain, medium chain, long chain, and very long chain fatty acid. Now, coming to a concept called essential fatty acids. Usually, polyunsaturated fatty acids that contain double bonds beyond the 9th carbon atom. That means if a fatty acid has got double bond, 12th carbon atom, 15th carbon atom, or 18th carbon atom, so this type of fatty acids are referred as essential fatty acids, because we human cells don't have a capability to introduce double bond beyond the 9th carbon atom. So, any fatty acid that is necessary for us which contains a double bond more than 9th carbon atom, like 12, 15 or 18 carbon atom, simply we cannot synthesize them in our body. We have to take them in the diet. That means they become essential component or essential requirements in our diet. So that type of fatty acids are referred as essential fatty acids. So what are the essential fatty acids that are necessary for us here? So one of that is linoleic acid. As you see here, linoleic acid, it has got 18 carbon. So it has 2 double bonds, and look at the position of double bond in linoleic acid. It is at 9th and 12th position. 9th is fine. Our human cells can introduce double bond at 9th carbon atom, between 9th and 10th carbon atom, but human cells cannot introduce double bond between 12th and 13th carbon atom. So that 12 here, that makes it essential. Linoleic acid having double bond between 9th carbon and also 12th carbon, it is an essential fatty acid, and we classify that into omega-6 family of fatty acid. Always remember, so the omega-6, omega-3, all that we can understand from looking at where exactly is the double bond present in that particular fatty acid. So let me show you that by using this particular figure here. Okay, so if you look at the figure here, so there is a omega carbons. This is the omega carbon. CH3 carbon is the omega carbon, right? So if the double bond in relation to this omega carbon, if it is present, say this is omega 1, then we have omega 2, then omega 3, like that. If there is a double bond between omega 3 and omega 4, right? So we call that as omega 3 family of fatty acid, because in relation to the omega carbon, very first double bond is present at omega 3. That's what is omega 3 family of fatty acid. Now if there is no double bond here, say omega 4, omega 5 and omega 6, and there is a double bond. Imagine if it is like that. So very first double bond you are going to see at the omega 6th carbon. So that's why we will say that as omega 6 family of fatty acid. So the omega 3, omega 6, omega 9 family of fatty acids. So the indication is whatever the number that we say if we say it is omega 3 family of fatty acid, that means in relation to the omega carbon, that is number 1 carbon, W1, omega 1, omega 2 and omega 3. So omega 3 carbon, there is a double bond here at the omega 3 carbon. That's what is referred as omega 3 family of fatty acids. So you got to be aware of that. So sometimes it will be required for us to determine a fatty acid which belongs to either omega 3 or omega 6 family of fatty acid. So you'll have to identify that. So that's where this will be useful. So let me take you back to linoleic acid. So the linoleic acid is omega 6 family of fatty acid. How do you find out linoleic acid is omega 6 family of fatty acid? For that you will have to look at this numbering system here. So number 18 indicates it's a total number of carbons present in that fatty acid. Number 2 indicates total number of unsaturated double bonds or the double bonds. So 2 double bonds there. And where exactly is the double bond? One is at 9th carbon, other is at 12th carbon. So how do you classify this linoleic acid into omega 6? All you need to do is take a total number of carbons, that number 18, and look at where is the last double bond that is shown in the numbering system. So the last double bond, it is at 12th carbon. So subtract that 12 from total number of carbons. So 18 minus 12, it is 6. So it is a omega 6 family of fatty acids. So this is the easy way of calculating or knowing fatty acids, unsaturated fatty acid. It belongs to which omega family. So remember that. So whenever we take linoleic acid in the diet, so this linoleic acid in the body, in our body, once it is taken in, it will be converted into arachidonic acid. And this arachidonic acid, it's a precursor of prostaglandins. I'm going to show you how exactly that will happen. Coming to another essential fatty acid here, it is alpha linolenic acid. Look at the numbering of alpha linolenic acid. It is 18 carbon total, 3 double bonds are there, and the location of double bonds is 9, 12 and 15. So that means 18 carbon, total number of carbon is 18, and the last double bond is present at 15th carbon. So subtract that number from 18. So 18 minus 15, you get 3. That means this is omega 3 family of fatty acid. So alpha linolenic acid is a omega 3 family of fatty acid. So example for other example for omega 3 family of fatty acids other than alpha linolenic acid, which is coming from plant oils. It is 𝐼𝐼𝐾osa-pentaenoic acid and 𝗏𝐞osa-hexaenoic acid. Look at the numberings of 𝐼𝐾osa-pentaenoic acid and 𝗏𝐞osa-hexaenoic acid. You can also find out whether they are omega-3 or not. So look at 𝐼𝐾osa-pentaenoic acid. It has total number of carbons as 20. 5 double bonds are there and they are present at 8, 5, 8, 11, 14 and 17. So the position of last double bond is at 17th carbon. That means 20 minus 17. So you get 3, that is omega-3. Similarly, look at 𝗏𝐞osa-hexaenoic acid. It has 22 carbon. Position of last double bond is 19 here. So 22 minus 19 is 3. That's why it is omega-3 family of fatty acids. So like this, so we can find out easily it belongs to which omega family of fatty acid if you know the numbering system that is mentioned along with the fatty acid. Now, this alpha linolenic acid, 𝐼𝐾osa-pentaenoic acid, 𝗏𝐞osa-hexaenoic acid, so all of them when they become component of our cell membrane, because they are polyunsaturated fatty acids, they will increase fluidity of the membrane, and also they are precursors for eicosanoids. These are inflammatory mediators that are necessary to take care of certain inflammatory agents that are getting into our body. So here is the figure that will show you how these essential fatty acid will help us in fighting against infection or for inflammatory process. So here is the membrane phospholipid that is containing any of these essential fatty acids and their derivatives. Like arachidonic acid. Arachidonic acid is a derivative of alpha linoleic acid. And so, whenever there is an inflammation, at that time, a phospholipase A2 enzyme, it will be activated and it's going to release arachidonic acid into the cell. And arachidonic acid is a omega-6 family of fatty acid, and this will get into inflammatory pathway. That is, there can be two types of inflammatory pathway. One is a lipoxygenase pathway. Other is cyclooxygenase pathway done by COX enzyme. Okay, so lipoxygenase pathway you get leukotrienes, lipoxins, whereas in cyclooxygenase pathway you are going to get prostaglandins, prostacyclins, thromboxanes, and all of these molecules, they are pro-inflammatory in nature, so they will lead to acute inflammation. And if this acute inflammation is not resolved in time, it will go into chronic inflammation. That means arachidonic acid derivatives, which are omega-6 family of fatty acids, they all will produce pro-inflammatory molecules. That means they are going to increase pain, fever, temperature, and then rubor, calor, dolor, all that kind of inflammatory signs will be increased in the presence of arachidonic acid derivatives, usually done by cyclooxygenase and lipoxygenase enzymes. Note that this cyclooxygenase enzyme can be inhibited by NSAIDs, that is non-steroidal anti-inflammatory drugs, and also aspirin. They are going to inhibit cyclooxygenase enzyme. There can be two types of cyclooxygenase, that is cyclooxygenase number 1 and cyclooxygenase number 2, COX1 and COX2. I'm going to go into little more detail on that in the next slide. Okay, now steroids. Whenever patient is treated with steroids, the steroids directly inhibit phospholipase A2 enzyme, thereby do not allow release of arachidonic acid. That means prostaglandins and leukotrienes are not made, thereby pro-inflammatory molecules are not synthesized, and that will decrease fever, that will decrease swelling, that will decrease pain and redness, edema. All these things can be decreased by prescribing steroids, which will basically inhibit phospholipase A2 enzyme. Coming to omega-3 family on the other side, omega-3 family fatty acids, 𝐼𝐾osa-pentaenoic acid, 𝗏𝐞osa-hexaenoic acid by lipoxygenase pathway, all of them, they will go into resolvin formation, resolvin protectins, mericins. All of them, they are pro-resolving anti-inflammatory cytoprotectants. So these pro-resolving anti-inflammatory cytoprotectant molecules, they do the resolution of inflammation, thereby they basically keep homeostasis. So inflammation is a good thing, but it has to be controlled in time. If it goes overboard, inflammation becomes too much, that becomes problematic. So it has to be controlled. So omega-6 and omega-3 family of fatty acids need to be in optimal concentration, so that inflammation is kept in a homeostasis. So pro-inflammatory and anti-inflammatory molecules should be properly controlled there. Now, some of the inhibitors of inflammatory mediators, like as I already told you, arachidonic acid released from membrane lipids done by steroids. Now, arachidonic acid going into COX pathway, which is mediated by COX-1 and COX-2. COX-1 is a constitutive enzyme. It is expressed by small intestine, expressed in the stomach. So, it is basically maintaining the necessary requirement for inflammation so that things are taken care. Like as it is shown here, COX-1 by increasing PGI2, prostaglandin I2, it causes gastric protection by increasing gastric secretion, especially hydrochloric acid, bicarbonate and all that. It is controlled. Mucus secretion is done here. Thereby, gastric protection is done by COX-1 through PGI2. Now, thromboxane A2, it controls platelet function. Now, whenever traditional NSAIDs, that is non-steroidal anti-inflammatory drugs are prescribed, like aspirin is prescribed, what it does, it's going to inhibit both COX-1 and COX-2. COX-2 is an inducible enzyme usually done by macrophages, and we should target COX-2 enzyme because COX-2 enzymes are basically, they cause this against inflammatory stimuli, like it may be trauma, interleukin 6, interleukin 1, interleukin 8, all of them, they basically induce this COX-2 enzyme, and COX-2 enzyme through PGE2, prostaglandin E2, it causes pain, it causes fever, and also through BMP superfamily, it is helpful in bone formation also. That's very important there. So, when you inhibit COX-2 in order to take care of pain and fever, so we also have a additional kind of side effect there. It is going to decrease BMP superfamily of proteins, thereby decrease bone formation. So, constant intake of this type of non-steroidal anti-inflammatory drugs may also have an effect on bone formation in our body, although they are able to control pain and fever. And other than that, since non-traditional, like traditional NSAIDs, they are not only inhibiting COX-2 enzyme, they are also inhibiting COX-1, thereby gastric protection can be taken out. So, that means it can lead to gastritis or increase in the acidic environment in the stomach, because that mucus secretion can be decreased, which is generally in response to COX-1 through PGI2. So, that's why traditional NSAIDs like aspirin may lead to gastric irritation, leading to increased gastric acid secretion, a decreased protection that can lead to gastric ulcers. Okay, not only they will have effect on bone formation, they also have a deleterious effect on our protective layer of the stomach, which will lead to gastric ulcers. Okay, so now, what about selective COX inhibitor, like COX-2 inhibitor? COX-2 selective non-steroidal anti-inflammatory drugs like celecoxib, rofecoxib, all this kind of coxib group of drugs, they selectively inhibit COX-2 enzyme only. They don't inhibit COX-1 enzyme. So, by selectively inhibiting COX-2 enzyme, they are going to efficiently decrease pain and fever, although they have an effect on bone formation, but this gastric protection is maintained there because we are targeting only COX-2 rather than both COX-1 and COX-2 enzyme through traditional NSAIDs. So selective COX-2 NSAIDs, so they are going to block only COX-2 enzyme and its pathway there. So remember about that. So steroid part I have already explained to you. Steroids inhibit phospholipase A2 and thereby effectively blocking cyclooxygenase pathway and lipoxygenase pathway and control inflammation there. And the COX-2 inhibitors, so there are two types. We have cyclooxygenase 1, cyclooxygenase 2. So non-selective COX-1 and COX-2 inhibitors, usually these are high dose aspirins, ibuprofen, diclofenac, naproxen. So basically they inhibit both the enzymes and thereby they may have some side effects. Whereas selective COX-2 inhibitors like coxibs, they specifically inhibit COX-2 enzyme, thereby they efficiently decrease pain and fever rather than leading to any gastric ulcer formation. So, have an idea on that part also. Coming to number of atoms and number and position of common double bonds present in different types of fatty acids, just as an example. So, we have 16 carbon, one double bond present at 9th carbon atom. So, we have palmitoleic acid present in nearly all fats. Then we have oleic acid, elladic acid, linoleic acid. Linoleic acid we have seen there. 18 carbon 2 double bond present between 9th and 12th carbon atom. Linoleic acid is an essential fatty. And now the linolenic acid. Linolenic acid, alpha linolenic acid is another essential fatty acid which belongs to omega-3 family of fatty acid, which is essential for us for all the anti-inflammatory mediators, pro-resolving agents that we need them. Arachidonic acid we can get that from linoleic acid in our cells. Whenever we consume linoleic acid, it can be converted into arachidonic acid and it will become part of our cell membrane, and that will help in formation of prostaglandins in our body. Now coming to 20 carbon fatty acid, that is icosapent pentanoic acid with a 5 double bond, and docosahexaenoic acid having a 6 double bond. So these are omega-3 family of fatty acids, and they are abundant in fish oils especially, and they are participating in protectins, myrcene formation, and they are anti-inflammatory, pro-resolving molecules which will keep the inflammation under homeostasis. Now coming to other special type of fatty acid which has got a branches in the hydrocarbon chain, referred as branched chain fatty acid. Now fats coming from ruminant animals and dairy products, they contain a significant quantities of branched chain fatty acid, and example for branched chain fatty acid is phytanic acid. As you see here, there is a carboxyl group COO- here, that's number 1 carbon, then number 2, number 3 carbon has a methyl group attached, which is unusual. So normally fatty acid should have carboxyl group COO- and then CH2, 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 CH2 like this, and end as CH3, that's a methyl end. Whereas here there are CH3s which are attached to the hydrocarbon chain. So this type of fatty acids we call them as a branched chain fatty acid, that you must be aware of. So now the branched chain fatty acid, when they become part of our membrane and also part of our body, whenever they need to undergo cell turnover, when the cell turnover happens and when the branched chain fatty acid like phytanic acid has to be oxidized, and the phytanic acid is initially oxidized in the peroxisome and later it is continued in the matrix of mitochondria. So if there is any defect in the oxidation of phytanic acid in the peroxisome, it will give rise to a disease called Rebsum disease, and Rebsum disease leads to accumulation of phytanic acid in the plasma and tissues. Remember that point also. Coming to briefly some of the properties of fatty acids. So the first property is solubility. So lipids are generally not soluble in polar solvents like water, but they are soluble in non-polar solvents, because non-polar solvents are they keep hydrophobic molecules like hydrocarbon chain soluble there. Melting points. Now as we have seen with the table earlier, the melting point of saturated fatty acid is generally higher compared to unsaturated fatty acid. So higher the number of double bonds, so lesser the temperature that is required to keep the fatty acid soluble state. Now coming to hydrogenation. Hydrogenation is the conversion of unsaturated fatty acids into saturated fatty acids in the presence of a catalyst like nickel. So basically it's an hardening of the fatty acid, hydrogenation process. Now the halogenation process is reaction of unsaturated fatty acid with the halogens like iodine number. So halogen like iodine can be used to determine the level of unsaturation. So iodine number determines the extent of unsaturation in fats and oils. So higher the iodine number, higher the number of double bonds present in a fatty acid. Remember that. And then the oxidation and reaction of unsaturated fatty acid with the molecular oxygen, it will lead to formation of corresponding peroxides, epoxide, aldehyde forms of fatty acids, and it will change the odor of a fatty acid. Fatty acids generally become rancid by this type of oxidation, and the degree of this rancidity, that is oxidation of unsaturated fatty acid, degree of unsaturation which is degree of oxidation which is referred as rancidity, it can be measured by something called as acid number. That means higher the acid number, higher the rancidity. And how do you prevent rancidity or oxidation of fatty acid? It will be done by antioxidants like vitamin E. So higher the levels of vitamin E in the oil, it will prevent rancidity or oxidation of unsaturated fatty acids. So this is all about different types of fatty acids that we have, and we have broadly classified them into saturated fatty acids and unsaturated fatty acids. And unsaturated fatty acids, furthermore, we have classified them into monounsaturated and polyunsaturated fatty acids. And also we have classified fatty acids based on the number of carbon atoms in them as short, medium, long and very long chain fatty acids. And also we have seen some of the specific fatty acids and their importance, including essential fatty acids and how they participate in membrane formation and how they will participate in inflammatory mediation and anti-inflammatory process. So that's all about lipid chemistry, that is depicting more of about a classification of fatty acids and their applied aspects. Thank you.